Hi everyone. It's a pleasure to have you all here. Can you hear me? Because we're having technical issues. Could someone type in the chat box if you can hear me or not? Because apparently Gord can't hear me.
I can't hear you. Nope.
Okay. The others can. The
I'm
world
not sure
can.
what to do. Do you want to reboot or should I? I mean, I look like I'm fine. You can see and hear me fine, right? I'm not sure if anyone else can hear you. I'll mic myself off.
Okay. We're having a glitch where Gord cannot hear me, but I can hear him, and I guess everyone else can hear us both. So welcome to everyone. It's a pleasure to have you here, morning, afternoon, evening, wherever you are in the world. It's a real pleasure to introduce Gord, who's done seminal work in understanding interneurons, their development, and their integration in the cortex. So Gord did his PhD in Toronto, following his PhD. So at the University of Toronto, and he then went to a postdoc with Mary Beth Hatton working on the cerebellum at Columbia University, and moved with Mary Beth to Rockefeller, where he then, from postdoc, became an independent staff scientist, a non-lab head PI, and then moved to NYU to head his own lab at NYU. He in his own lab. He started studying cortical progenitors, and their development, integration in the cortex. He then became interested more specifically in interneurons, and his lab has been working on that ever since. And has, as I said, made seminal contributions on the subject. Gord actually also really built up the NYU neuroscience program. He did a fantastic job doing that, recruited some great people, and more recently moved his lab to Boston, where he basically straddles the broad at MIT and Harvard. And where his labs work on different but complementary things that I guess we will hear about today. So I first actually met Gord under very interesting circumstances, which were simultaneously exciting and dubious. Where Gord, myself, Frank Polleux, Dietmar Schmucker went to a rock concert by Pierre van der Hagen. And a series of events that I'm not going to divulge to an international audience in recorded form occurred, in which Gord and I simultaneously risked and saved the life of Frank Polleux. And I'll let Gord decide how much of that story he wants to share. But it was an unforgettable experience, and Gord and I have been in touch ever since. And so, Gord, it's over to you. It's a great pleasure to have you, and we're all looking forward to your talk. Go ahead.
Okay, hopefully everyone can hear me. Bassam, can you kill your mic so I don't get an echo? Okay, hopefully this will work, and we are launched. Bassam, just tell me if that everything's working, and I will get going. Yes, Bassam? Okay, I can't confirm that anyone can hear me. Can you guys, maybe someone just send me a message if that all seems to be working, because I don't want to talk to myself for half an hour. We cancer you. Okay, go ahead. I guess we're good. Okay, it's really a pleasure to talk at this forum. It's kind of sad that when I get together with the largest crowd I've been with in months, I can't hear or see any of you. So I'm looking forward to the question and answer period. I will take my glasses off because I've realized over many Zooms I look slightly cyborg when I do that. So I'm really excited to tell you a bunch of new data from my lab, none of which is published, and really would love to get your feedback on this. So I'm going to talk about two of my favorite cell types, or at least cell groups, both of them interneurons, parvalbumin and somatostatin. Those are two of the major cardinal classes of interneurons, and something my lab's been fascinated with for many years. So what most of you probably appreciate is the brain is not at birth fully functional, and that young children go through a initialization period where they begin to learn about the world, something we all know if any of you have had children. So initially, the world is new and novel to the young child and is transcribed accurately to the brain. It's basically a period where the world is informing the brain of the outside world. And at some point, we go from this initialization period to a period when the child grows up, and now the pattern of the world is compared to the pattern of the brain, and we use prediction coding to try understanding what the world is doing and whether we're doing the right things in the world. And that's a process of our brain talking to the world through efferent copy, and the world talking back to the brain. And so that transition is something that I'm growingly interested in. And I personally, and I think many of my colleagues are convinced, has something to do with the way interneurons function. So here's a view of interneurons in the cerebral cortex taken from a review I did with Adam Kepesh. And here you can see the basic classes. And each one of them is unique in the way they talk to neurons. VIP and somatostatin tend to talk to the dendrites of neurons. PV and CCK tend to talk to the soma, and they actually talk to different neuronal populations. So while most of the interneurons are talking to excitatory cells, the VIP cells are unusual in that they're talking to inhibitory cells. Today, I'm just going to focus on two of these four major classes, the somatostatin and parvalbumin ones. They're really interesting to study together for a couple of reasons, one of which is they're the most prevalent interneuron types in the brain. And secondly, they are quite different. And even though they come from common progenitors, they turn into two very, very different cell types. And so really what we've been trying to understand is how do you make two different cell types from a common progenitor? And then how do you integrate them in the brain so they can do different things, not only with one area in the area of the cortex, but across areas of the cortex. So today, my talk is going to look at both aspects of that, and hence the title, both nature and nurture. In the first part of my talk, I'm going to talk about intrinsic factors, the transcripts in terms of genes that are expressed in the cells, and some newer work where we've looked at the chromatin structure of PV and SST cells longitudinally across development. After that, I'm going to kind of switch over and talk about extrinsic factors. And by this, I mean the afferents that come to PV and SST cells. And I'm going to begin that by giving a survey of the connectivity of PV and SST cells in different parts of the cortex, and then talk a little about the development of how they occur and some ideas we have about how PV and SST might be differentially affected by activity and intrinsic genetic components. And of course, all of these talk back and forth to each other. So the real challenge is trying to understand how nature interacts with nurture over development. So the first part of the talk, as I said, is going to be on chromatin, and the last part of the talk will be on extrinsic inputs. And in that second part of the talk, I'm really going to focus on different areal territories. And there I'm really going to compare sensory areas, S1 and V1, to associative areas, and in this case, the M2, which is sometimes referred to as the anterior lateral motor, or ALM. But let's start with the intrinsic part of the talk. And this work began really with an absolute breakthrough technology that my colleagues Steve McCarroll and Evan McCosco, along with Aviv Regev, came up with in Boston. And that was this single cell transcriptomic analysis. And I was lucky enough when Rahul Satija, who worked with Aviv and Steve, came to New York, and we started a collaboration to try understanding the developmental trajectory by which interneuron diversity appears in development. So as I'm pretty sure everyone knows by now, you can tell interneurons and excitatory cells and pretty much any cell in the body based on their transcriptomic signature in the adult. And here's a picture from that adult comparing, I think this one's actually maybe from the alum or ours, but it all looks fairly much the same, where you can see that diversity of interneurons in the adult cortex, and you can see they're divided into those 4 cardinal classes I mentioned. And so back in 2018, with my former postdoc, Chris Mayer, and Rachel Bandler, a former graduate student, along with Rahul's lab, we decided to understand longitudinally how that diversity appears over time. And what you can see here is one of the basic messages of that is that initially you can see only the grossest divisions, the cardinal classes, and that over time those classes become ever more refined until you see the diversity that you see in the adult. So again, I'm only going to concentrate today on the PV and the SST ones for the reasons I talked about. And one of the reasons why they're such a great pair to put together, even though they're ultimately very different, is that they all start off in this progenitor structure known as the medial ganglionic eminence. And in terms of that medial ganglionic eminence, one of the real surprises we got from our RNA analysis was we weren't sure when exactly we would start seeing cell type diversity. And within the MGE, you're essentially having cells in 2 fundamental states: this dark gray area, which is the proliferative zone where they're dividing, and this post-mitotic area where they kind of leap out of proliferation, never to return, and start their long migration up to the cerebral cortex. And this story is really been piloted by the wonderful work of Kate Alloway, doing all the wet work and some of the computational work, and Mariano Gabito, who really is a wonderful computational biologist, who I'll bring into the story as we get to the more computational part of this. So in early postmitotic neurons, and this is going back to the work with Christian Mayer and Rachel Bandler and Rahul, we realized that, to our surprise, that in mitotic cells, we could see very little evidence of cell type diversity. We could see in a general way they were probably GABAergic, but not much more in terms of their character. And what we realized in that analysis is that as soon as they become postmitotic, you can see some level of diversity. Not the diversity you see in the adult, but you can see 3 branches, and you can see that here. So the red is the more immature cells in this diffusion map, and those green tips indicate branch 1, branch 2, and branch 3, which when you look at the gene expression in them clearly are different cell types, at least in general categories. And most of our time is spent on this branch 1, which are all the interneuron classes. So somewhere from going from mitotic to post-mitotic, transcriptomically, you can at least see which ones are the interneurons versus the projection cells. And since we were puzzled by this fact that the RNA didn't show any evidence of mitotic commitment, we wondered if we were missing a signature. And the place that signature most obviously could be was in the chromatin. So for as long as people have been doing molecular biology, the question of DNA, RNA, and protein has been one that's captivated people's attention. And what we know in terms of gene regulation is you can look at now at a single cell level the transcriptomic diversity by looking at the different RNAs. But now increasingly, we can even look at the chromatin diversity at a single cell level because of the advent of a new technique that allows you to look at heterochromatin versus euchromatin, and basically be able to look at chromatin that's highly compact, where the genes are off, and then basically identify areas where genes start opening up as histones get released and transcription starts occurring. And we can see this at a single cell level because of the fantastic technology that was pioneered by the Greenleaf and Greenleaf lab at Stanford and Howard Chang. And because of a method called ATAC-seq. So ATAC-seq, other than obviously being the initials of the nucleotides, stands for assay for transposon accessible chromatin. And so the way it works is the transposase accessible chromatin. And so the transposase goes in and cuts the DNA wherever histones aren't tightly packed, and then it inserts primers that you can amplify up and at a single cell level distinguish peaks that are open chromatin versus closed. And so when you do this in the adult, you can get a pattern of the chromatin signature and compare it to the RNA signature. And as shown by Bing Reng and others, it turns out in the adult, either method can give you that identity at the single cell level. So you can tell what cells are which, either by looking at the RNAs in those cells, or you can look at the pattern of accessibility in the chromatin, and those two can be aligned. And so in the adult, they match rather nicely. And so we decided we'd start back in early development and see how that pattern develops over time. And so our initial hypothesis that I already mentioned to you was the idea that in early development, perhaps we could get a hint of the cellular diversity at a chromatin level, which wasn't readily evident at the RNA transcript level. And so that's what we started off trying to look at. And as you'll see pretty quickly, that turned out not to allow us to get further insight. So focusing just on these MGE cells, what we wanted to do is compare mitotic to post-mitotic cells. And as any of you in the interneuron field probably know, DLX is a wonderful post-mitotic marker of all the interneuron and other GABAergic populations, but it's not on in the proliferative cells. And so from some early work from a postdoc in the lab, Orly Wapinski, she sorted these cells out into DLX positive and negative, and then Kate set forth to start comparing the single cell RNA-seq signature to the single cell ATAC-seq signature. And so here, with the maturation trajectory that we can do with these diffusion maps, you can see that they both have these single trajectory going from mitotic to post-mitotic. But what we didn't see any more clearly, and as I'll show you actually less clearly, was that the chromatin signature provided any more resolution of cell types than the RNA transcriptomic signature in the mitotic cells. And in fact, in both of them, they look like pretty much one uniform population. But as I told you before, as soon as they become post-mitotic, we see the emergence using diffusion map analysis of these three basic branches, an interneuron branch and two projection neuron branches. And so the question was, what does ATAC-seq tell us about the chromatin signature? And what you can see here, at least crudely, is those three basic branches are there, but you'll see they're less developed at this stage. And what you can see is a whole lot more brown cells, which are the mitotic cells. And since these are the same sample looked at it two different ways, what it indicates is the ATAC-seq signature gives a lagged signature of the cell type identity, which follows but does not lead the RNA-seq signature. So here you can see a bunch of genes and their expression profile starting in the mitotic cells, early post-mitotic, and slightly differentiated cells. And you can see the turn on of genes indicative of those various states. And with the ATAC-seq, we see a similar signature, but you can see when you're looking at the accessibility of those loci, it actually seems to lag on. So the initiation of branch selection seems to be more a product of turning on RNAs, but it isn't baked into the chromatin. Somehow those genes turn on, even though it's not obvious that the accessibility of genes that are turned on is all that different from genes that are not yet turned on. And when we dug down in that and looked at it, and looked at genes that were really good branch markers, and these are MAF-B and TCF4 for branch 1, EBF1 and MICE2 for branch 2, or ZIC1 and NKX2 for branch 3. We can see the transcriptomic signatures of each one of them. And then we decided to go look at those loci. And so these are enriched motifs that are targeted by these genes. And what we can see is that these genes in branch 1 and the interneurons are highly enriched. So these transcription factors are presumably targeting a whole lot of genes that bind these two transcription factors. Similarly in branch 2 and branch 3. And this enrichment is actually really marked. So the least pronounced one of those is MAF-B, which is has a p-value of 10 to the minus 13th, and that goes up as high as 389 to the minus 389 with mice 2. So these motifs are highly enriched within these 3 branches, even though the signature of cell types is still fairly faint. So what happens next? How do these cells go from the state I just showed you here, where the attack-seq and the RNA-seq gave very different levels of resolution up to this P28 mature level, where those two align beautifully. So what we did was start moving along and start looking at various time points and try comparing them. And so the problem, of course, is how do you align the RNA-seq and attack-seq to get a direct measure of how they align nicely? And Rahul Satija, really a wonderful computational biologist, real pleasure to collaborate with him, has come up with this method where you can use canonical correlation analysis to line up the attack and RNA-seq, and that you can assign a number to. So if you have a high correspondence between the two, we'll give that a number of 1. And if the RNA and the loci have very low correspondence in the loci, that approaches 0. And so when you compare this in the adult, as you might expect, based on the beautiful alignment of these two, the peaks are all up here around 1. But as you go backwards to P10, that's slightly less pronounced, but then really starts falling apart in this E13 and E18 time point, where you can see the alignment of these two is far less distinct, and you really can't align the attack peaks and the RNA-seq peaks very well. So why is that? Why is it that these align so poorly at this time? Because we know that we can see gene transcript scores. So what is it about the chromatin motifs that make it hard to look at? And so we looked at a few candidate molecules. Here's alpha-1 as an exemplar. And you can see even by E18, it's much more strongly expressed in the SST cells than the PV cells, and that just gets more pronounced as time goes on. But if you look over here at the accessibility peaks, well, you really couldn't tell based on the chromatin organization, the alpha-1 organization in the PV and SST. And that's true of a number of other loci. All of these are genes that allow you to see PV and SST, these two PV and this SST from each other developmentally. But you can see here that developmentally, their chromatin signature in terms of accessibility isn't very different. And it's only as you get into these more mature phases, those differences become more pronounced and easier to see. So we wanted to use this Jacquard method to kind of globally compare how chromatin organization changes over time. And the Jacquard method allows us at a global level to generally see how chromatin compares within a time point and between cell types and across time. And what you can see from this Jacquard plot is that at early developmental times, PV and SST look very, very similar as compared to later developmental times, whereas they become more and more distinct as things move on and develop. So to drill down on that a little more, we looked at particular peaks and started looking at some of our favorite genes and asked how those loci transform their accessibility over time as development proceeds. So the branch markers, as I told you, are very distinct in branch 1 versus branch 2 and 3. You can see a nice branch marker for both PV and SST as early as E14. And as you get into the adult, even though these are clearly still interneurons, those branch markers diminish, and as presumably other genes take over and become more important. Similarly, interestingly, you can tell PV and SST apart as early as E14 and certainly by E18 by some of these migration markers and developmental markers, which transiently allow you to tell these apart. But again, those genes become less distinct in the adult as more differentiation genes kind of take over from these early differentiation genes. And finally, here you can see that specific genes, in a general sense, become PV and SST specific as you get into adulthood. And when you go down to look at specific motifs, and so this is now the adult equivalent of what I was showing you with the branch markers, we can see that MEF genes, which we had previously shown are really important for PV cells, really target PV specific genes. So a lot of the genes that are specific to the PV cells have MEF binding sites, both at P10 and P28. And a novel finding, which we hadn't seen previously, is that PAL and OX sites become highly enriched in SST cells as maturation progresses. So it's really interesting how early on and late on specificity is really dictated by different transcription factors. But as you might expect, transcription factors really drive cell type differentiation genes, particularly in these MEF and POW ones as you get into adulthood. So what about a more global view? And this is where the computational help of Mariano Gabito and Giuseppe Saldi became really, really promising. This is what's called a gene regulatory network, and we did in collaboration with the Bonneau Lab, a lab which I share both Giuseppe and Mariano with. Rich Bonneau came up with this idea of a gene regulatory network by taking priors of which transcription factors target which genes, kind of how we did with the PEF and MAO gene, the MEF and POW genes. And to take a first pass look at this, you might conclude, oh, the network for immature cells is much more complex than the network for mature cells. But this is taken from a very specific perspective. We're only looking at shared edges here. So what we're looking at is at E18, how many of the transcription factors shown in red have common targets early in development, and here at a later time, how many transcription factors have common targets later in development. And what you can see is the PV and SST cells share a much more common network early and late. But that kind of goes counter to intuition. We would think that as they differentiate and become more specific, this network should be more complex. And then if you pay attention to the, and I just want to say that this work was really the work of Mariano Gabito and Giuseppe Saldi. And I just want to give a particular shout out to Mariano. He's about to move on from his postdoc, just a fabulous computational biologist, worked with Charles Zucker, Larry Abbott, and Tom Jessel, and did great work with them. And he's been an absolute pleasure to work with here. So if you're looking for someone who can bridge between computation and neuroscience, he would be a great choice. And he's on the job market. So I would grab him while he's hot. Okay, so I made the point that the shared regulatory network, as you can see in the previous slide, looked much more shared in the E18 than the P28. And here you can see it at a more statistical level. And so you see that just like the network showed, the number of shared edges is much higher in the E18 than the P28. However, that's not to say the network is less complex at P28. If you start looking at the new regulation, the unique regulations in each network, what you can see is that, well, here only 40% of the things are still shared at E18, and that's the network I just showed you. By P28, 80% of the unique edges are specific to PV or SST. So only 40% of these at E18 are unique. In these two, it goes up to 80% by the time you get to P28. And if you actually look at the number of total edges, and this is why we didn't show you the whole network, they're up around 20,000 for either SST or PV by the time you get to P28, and they're only around 10,000 when you're back here at E18. And so what that says is that these two cell types, which come from a common origin, have a lot of shared edges and are interneurons, and they're basically getting themselves started as a shared cell type. But as they differentiate and start doing distinctly different things and have unique genes from one another, we get distinct networks. And suddenly many, many more genes get turned on, 20,000 instead of 10,000, and that the edges that are specific to PV or SST become much more selective to one or the other. So I'm going to summarize this part of the work before going on to the second half of my talk. What we saw when we looked at the ATAC-seq was that early embryonic ages, what we had found with RNA-seq gives you a much more precise and an earlier readout of cell types. And at those times, the chromatin, it seems much more plastic and much less baked in. By postnatal times, ATAC and RNA-seq start to converge and start looking to each other. And when you think about what that may mean, it basically says that the cell fate plasticity is probably most accessible by looking at the chromatin, but the cell fate state is better looked at by looking at the RNA. So this analysis is still ongoing, and I just want to give you some hint of things we're doing now to get a better feeling of the contribution of chromatin to development. So The GRN analysis, literally the stuff I showed you in the last couple slides is really only a couple days old. And we're doing a lot more analysis to try understanding how the GRNs progress and develop over time and to validate it. So how do you go from a computational prediction to a validation? And the obvious way is to stress the system. So we're going back and removing transcription factors to try understanding how that changes the GRN. And you can do this iterative approach of removing a gene and seeing how the network changes. We're starting this with MEF2C. For those of you who followed my work previously, you know that in our transcriptomic analysis, we identified MEF2C as an important gene for PV differentiation. If you were paying attention in the last few slides, you can see that MEF motifs are important, particularly in PV cells from early development, and that continues into adulthood. So we're going to do a loss of function with MEF2C and see how that affects the RNAs as well as the chromatin structure. And finally, you always want to do these things orthogonally. And so for the work I was telling you about POW and MEF sites, we're doing cut and run, which is an independent way to really make sure that the things we think are binding MEF are really binding them, and the site within PV cells and the things we think are binding SST and SST cells are binding them. And all of this will hopefully allow us to get much better at doing these gene regulatory networks. And the idea is that we'll get to a point where you can actually make predictions about what's under the hood of how a cell is functioning and really how different genes are regulating it. And that's important not only for development, but because it probably will be super important for disease. So one of the genes we saw was TCF4. It's a gene involved in a specific monogenic disorder. Wouldn't it be great to know exactly what it's regulating in interneurons and to what extent the disease can be predicted by its function in, let's say, PV cells on its own? So that's the future. Okay, for those of you who are more interested in the assembly of circuits, we're going to switch pages now and go entirely from talking to chromatin to talking about the development of presynaptic inputs in these same cell types, the PV and SST cell types. And one of the things that we really want to emphasize here, and it has been really a puzzle since it was revealed a year or two ago, is how do PV and SST cells integrate within different functional areas? So work by Mike Stryker, Massimo Scanziani, Barry Connors, and my colleague Bernardo Rudy have looked at how things like VIP cells incorporate in M2 versus S1 versus V1. And the lesson that really has come out of that is that they seem to make canonical circuits in each one of those. But this is a little puzzling when you think about it, because really S1 and V1 are sensory areas that are trying to see the world and imprint it on the brain, where M2 is really taking higher motor function and assembling it into motor assemblies that are getting the world to act back out on your muscles, your brain to act back out on your muscles. And how do these two cell types, which are ubiquitous in both these areas, differentially function here and here? And so we decided to tackle that, or I should say more specifically, a fabulous postdoc, Gabrielle Pouchelon, decided to tackle that by looking at how the connectivity, and in this term, the afferent connectivity of PV and SST cells is assembled not only within sensory areas, but in sensory and association areas. And I really have to say that it's a particular pleasure to present her work in this form. She was the first graduate student of Denis Jabadon, who really started this series and had two wonderful Nature papers with them. And so now I think you'll see what her next chapter is, and I'm going to show you the work she's done in my lab over the last 5 years. So one of the things we know about cortical organization is, and it comes from that first slide I showed you, is that the cortex, first and foremost, is programmed by the periphery. And though that information is taken through the thalamus in these barreloids and then imposed in these barrel fields up in the cerebral cortex. And those of you who know my wonderful colleague, Gia Lopez Bendito, knows that she did beautiful work in terms of showing that the activity pattern within these barrels is matched in the thalamus and the cortex. And more recently, in this wonderful work with Noah, Anton Balonos, was able to show that when the activity in the barreloids is perturbed, the barrel fields don't form properly. So it's actually imposing that information from the periphery on the cortex. Now, what most of you are probably aware of is this barreloid organization is first and foremost a product of the organization of the pyramidal cells. And what Gabrielle did with Denis during her graduate work was ask the question, to what extent does perturbation of this passive sensory inputs have an effect on the organization of the cortex? And so what she was able to show is that if you specifically target and actually kill the ventral basal thalamus, a little more extreme perturbation than was done by Gia, what happens is those barrels don't form. But what's interesting is if you kill them, rather than just silence them, then the higher order inputs, which normally are more restricted to S2, invade S1. And as a result, pyramidal cells in S1 take on the character of S2. So at this level, it looks like whether your primary sensory cortex or higher sensory cortex at the pyramidal level has a lot to do with what thalamic afferents you're getting. And if you can reroute the higher order ones to S1 instead of having them mainly restricted to S2, S1 starts taking on an S2 character in the pyramidal cells. So, okay, I live in an interneuron-centric view of the world. So I, of course, was immediately interested in how that was affecting the interneurons. And our thoughts on that were affected in a fairly pronounced way when this result came out from the Allen group back in 2018. And so this is not what I predicted, but is really, really a striking result. And that is asking, well, okay, how much do the interneurons versus pyramidal cells transcriptome vary across various sensory areas? And so what the Allen looked at were the interneurons in M2 and compared them to the interneurons in V1. And what you can see here in the summary diagram is that in M2 and V1, interneurons are practically identical in those two areas. However, pyramidal cells have marked differences of up to about 200 genes per cell type. And so transcriptomically, the interneurons look very similar in these two areas. And so this really puzzled us because how is it that you can have the same transcriptome, though presumably deal with much different information? You're certainly dealing with different pyramidal populations, but of course those very areas mediate quite different types of signaling in M2 and V1. And so what Gabrielle decided to do was to ask, let's just take a survey of inputs onto the interneurons in these two areas and ask two questions. How do they compare in the adult, and how do they reach that adult state? So I'm going to give you the take homes. There's a lot of detail here, so I'm going to be giving you a bit of a wild ride through it. But I want you to try keeping in mind some fairly global concepts that we think come out of this. And the first global concept is that the inputs, the afferents going to PV and SST cell, are actually in a general way more similar in terms of local and long-range intercortical connectivity, and to some extent subcortical connectivity in areas. In other words, if you're in M2, the connections for PV and SST in terms of afferents are rather similar and different from what you see in V1. The second principle we realized is even though the general connectivity is more or less the same in these areas, the order in which it's established is quite different. So what I'll show you is that, for instance, when local interneurons innervate PV and SST in one area is often out of sync when local interneurons innervate, when local interneurons innervate PV, it's different from SST, and similarly long range have a different timing in which these different populations are innervated. So the one thing I haven't really talked about, and probably is the thing most on your mind because it's what I brought up to start with, is what about thalamic afferents? And so there, again, both an areal and a cell type specific set of roles come into it. And what you'll see is that both in the order in which first order and higher order thalamus inputs target PV and SST, and in the abundance of first order and higher order inputs go to those cell types, is both area specific and cell type specific. So those are the three messages of what I'm going to try showing you. And then we're going to delve into a little about the nature and nurture by doing an activity versus genetic perturbation to see how it affects these. So let's first say, how do you go about this? How do you look in a broad scale way, all the afferents and the timing that target different cell types? And this question was really, to my mind, beautifully addressed by work from Wickersham and Callaway, where they used this modified Rabies method where they could label specific starter cells with Rabies and then monosynaptically label all their afferents. And so I'm going to assume most of you are somewhat familiar with this approach. It combines a AAV helper that uses genetic driver lines to target what specific starter population you want to get. And it basically does that by giving a receptor that allows the rabies to get in. And this isn't wild type rabies, it's pseudotype rabies with its MVA receptor. So it can only get cells with TVA. So you use genetics to put TVA in the cell type you want to query, and then you infect with rabies, and it only goes into the TVA positive cell type. So, of course, we want to do this in PV-SST, and in the adult, that's easy enough to do because they have great specific markers, PV and SST themselves. But in development, well, SST comes on rather early. Parvalbumin comes on rather late. And so that led to the question of how are you going to target parvalbumin cells long before the PV cells turn it on? And so Gabrielle came up with this intersectional strategy that worked just beautifully to target them. And that used the fact that we know that both these populations, PV and SST, have this gene LHX6 on it in both of them. But of course, the PV cells don't have SST and the SST cells do, and this comes on early. So we came up with a subtractive method where we could basically label all the cells that are LHX6 positive and then subtract away using the SST flip line that Josh Wong made, all of the ones that aren't PV. And so that allowed us at an early time through the selection away of SST cells to look at both PV and SST. And so this works really nicely. Here you can see the infected cells, and the yellow ones here are the starter cells, and the red ones are ones that connect to it. And then with a nice collaboration with Rosa Kozart and Yannick Ballman, we were able to both manually and computationally look at the population of starter cells and then normalize them to their connections to other areas of the brain. And so this was kind of a Herculean effort on Gabrielle's part, because she not only had to look at PV and SST cells, she had to look at them in 3 different cortical areas, S1, V1, and M2. And she also had to look at them at different times. So you can imagine how much work, that's like a 2x3 matrix. So she had 6 different things she needed to look at. And that doesn't even include the time points. So it was an awful lot of analysis. So you can imagine this data is very complex, and you can drill down for it for years. And I have a ton of extra slides for the aficionados if you want to get into it. But for the purpose of this talk, I'm going to just give you the 10,000-foot overview of what we learned. And so the first thing we learned was that local cortical connectivity, so that's the pyramidal cells and interneurons within a given area, are basically lower in M2 than in S1 and V1. So if you look at local connectivity, both PV and SST cells tend to have more local connectivity if you look at sensory areas than associational areas. They occur in both, but they're weighted towards local connectivity. However, if you look at contralateral connectivity, how one part of the cortex talks to the other, the pattern is opposite. And here, particularly in somatostatin cells, the contralateral connectivity is lower in SST than in S1 and V1 versus M2. And similarly, though, to a lesser extent, the contralateral connectivity to S1 and V1 PV cells is less than PV cells at M2. So another area where you see these differences in terms of the cholinergic cells of the basal forebrain, since we started doing this rabies, we were stared in the face something that we've known for a long time, which is that interneurons get a lot of cholinergic input, but it turns out that they get more cholinergic input if they're in M2 than if they're in S1 or V1. So both PV and SS2 get a lot of cholinergic input in M2, but the same populations in S1 and V1 get it, but at a lower level. And this is just showing you a picture of some of the double labeling. So here's kind of a low power view, which makes that point in kind of a heat map sort of way. And what you can see here is that the general connectivity of PV and SST in terms of cortical connectivity is matched across many, many different areas, but different across different areas. So here, by and large, PV and SST look similar in the areas that match and different, but it's more based on whether you're looking at S1 than you're looking at M2. And so you can see the sensor areas, by and large, have similar connectivity between PV and SST than the motor, this M2 area, but within them, they resemble each other. So here with the Pearson correlation, you can see the same thing, which again shows that diagonal line, which is saying PV and SST in any given area have similar global connectivity, and that changes depending on whether you're looking at V1, M2, or S1. So the general connectivity is similar. And remember, I want you to keep in mind two things. This is only looking at the adult so far, and it's also not really paying attention to the thalamus. And the rules of thalamic input are different, and I'm going to go through that in a minute. So let's first look at the developmental angle. So I said that PV and SST have similar inputs locally and contralaterally in terms of M2. So they both have more pronounced inputs from the contralateral than the local in M2, but it occurs on a different time scale. So if you look early in development, the local connectivity first gets in M2 to the PV cell and only later gets to the SST cell. And so you see with PV cells, they basically establish all their early connectivity very early and then only build on it slightly or maybe even decline it. Whereas the SST cell builds the local connectivity across development. In contrast, the contralateral connectivity first impinges on the PV, on the SST cell. You get very little contralateral activity initially, and then the PV cell catches up as time goes on. And similar rules occur in terms of subplate, as many of you may be aware of from work from Patrick Knold and Zoltan Molnar. The interneurons do get input from subplate, and this is something which we saw in our early rabies tracing from Natalia DeMarco. And basically, when you look at this more carefully, both PV and SST initially get quite a bit of subplate input, and that's most pronounced in S1, where you can see that unlike other areas, the SST cells get a huge amount, and that declines over time. And finally, another input which has been of particular interest to us is that we found in deep layers there is a transient input of SST into PV cells. So some work from a wonderful graduate student in my lab, Şebnem Tunçdemir, showed that the SST cells during development project to PV cells in deep layer, but those connections get weaker as time goes on. And similar results were published by my former postdoc and now independent scientist, Simon Butt. And so there's some sort of transient communication between SST and PV cells in deep layers that goes away over time. So that's an awful lot of results in terms of adult and development. So let me just summarize those results in the next diagram to give you the take-home messages of this. First and foremost, what I showed you is that in M2, you get less local inputs in the adult than you get in S1 and V1. And that's irrespective of whether you're looking at PV or SST. Similarly, so that's the same result showed for the SST. So that was the PV1 first and the SST1 second, and both of them have more local connectivity in S1 and V1 than M2. Contralateral, it's the opposite. In M2, the PV cell has more contralateral input if it's in M2 than it's in S1, and similarly, the SST cells have more contralateral input if it's in M2 versus S1. So the general patterns of local and long-range connectivity are predicted by area, but not by cell type. Now, what about development? What we see in development is that the local input of PV cells comes on before the local input of SST cells. So if you look early in development, PV cells have much more local input than SST, and then as time goes on, the SST cells catch up within M2. However, the contralateral input is first targeting the SST and only later do the PV cells catch up. So this may have to do, they're getting the information locally and long distance with a different time course. What about the subplate cells? So in all cases, irrespective of whether you're looking at M2, S1, or V1, there is strong subplate input to these interneuron populations developmentally, and they become much, much less in the adult. But strikingly, the area that gets the most subplate inputs is the SST cells, particularly in V1. So for whatever reason, that population gets an overabundance of subplate cells, and like everywhere else, they diminish over time. And finally, I told you about the connectivity developmentally, which we saw and is largely transient. And we had previously described it over here in barrel cortex, where SST transiently projects to PV. Turns out if we looked at motor cortex, it's even more pronounced. So even though these cell types are transcriptomically similar and have general long-range connectivity that's similar, the timing in which they're wired up is very particular to what area they happen to be in. Okay, I've saved the thalamic connectivity to last because really, as we said, developmentally, it's really the thalamus that first talks to these interneurons. And what we really were trying to look at is first order versus higher order thalamus. And so the question really here was, we know that there's this passive input that comes up from the periphery and basically gives you sensory input that you take in from your environment, and that goes through first order. But of course, that information also is integrated with higher order motor inputs so that when you're whisking actively, you can actually correlate what you're doing with the action you're taking with your body. And that tends to be a combination of bottom up and top down input, and that goes through the higher order. And so thalamic nuclei are nicely divided in sensory areas between first order and higher order, as are associational areas. And what we know is that these passive ones are the ones that really target the barrel, and the septal regions around the barrels are really higher order. So how do these interneurons wire up in these various areas? So here's kind of the result you get for the PV cells. Whether you look developmentally or in the adult, both the first order fibers dominate. So this is in the adult, and so it's like a 70-30 split in favor for first order fibers and higher order. And if we looked at P10 in development, the same pattern is true. What about the SST cell? Well, in the adult, the SST cell gets about an equal split of first order and higher order. But again, as you go back in development, they don't come in simultaneously. So by the time you're at P2, or if you go as early as P0, it turns out the first order comes in first and the higher order basically plays catch up over time. Now, it gives you the impression that these have totally different logic, where the SST first gets first order and then higher order, where PV always gets both. But this comes to the caveat that our genetic methods doesn't allow us to query these early time points of PV cells, because the intersectional genetics only works by the time you get to P10. So it's possible that if we went back earlier here and had a better way to look at PV, the same rules would occur. But even if they do, it's clear that the SST cells get the first order inputs earlier than the PV cells do, and ultimately get more of the higher order inputs than the PV cells. So this is kind of a summary of what I just told you. In development, first order inputs come in first and higher order inputs come in later. So if you look early in development, first order inputs dominate at P10 in both PV and SST cells. And then as you go into the adult, in the first order inputs are maintained at a much higher level for the PV cells, but the higher order inputs to SST cells increase till it's about 50-50. So all the analysis so far I've shown you just in S1, and from the first part of the talk, you're probably wondering what's happening in V1 and M2. And so it turns out, as might be expected, the rules for V1 and S1 are very, very similar. And so in both cases, the same pattern occurs: first order first order persisting in PV, and higher order coming into SST later. But when you look at M2, it's the same for the SST. Again, they have this balance of first order and higher order, but somewhat surprisingly, the PV cell actually gets an equal amount of first order and higher order when it's up in M2. So the wiring in terms of first order and higher order of PV cells is starkly different depending on whether you look at associational or sensory areas, where higher order inputs dominate or at least are equivalent to the first order inputs in M2, but much less in PV cells in S1 and V1. So Rabies is great to get a global idea of connectivity. What it's not great for is getting a global idea of strength. And the answer is we're not nearly as advanced on this part as we are on the connectivity question, but we have explored SST strength over time. And so this was first done by Şebnem Tunç-Demir when she was looking at S1, but now we've also looked at it in other areal territories of the cortex, and the rules seem fairly similar. And what you see there is early in development, these SST cells get strong thalamic inputs early in development, and as development goes on, those inputs get much weaker. And the other thing I talked to you is these SST outputs here are talking to PV cells, and here they're talking perhaps not at all to the PV cells, but much less. And so the strength is shifting along with the number. And of course, one of the things we really need to know and we're working on is how does the strength of the PV cells change over time? So one of the things we know from work by Carl Peterson and others is that in the adult, the PV cells have very strong inputs compared to the SST cell. And one of the things we want to explore is when does that strength come in? We know in general PV cell strength comes later, but exactly when it does, and is there any difference between first order and higher order? In terms of this, the tidbit I can add is initially we thought that in the shift, maybe it's the first order just getting weaker and it's somehow made weaker by the higher order. But we actually think that irrespective of whether you're first order or higher order early, you're strong, and irrespective of whether you're first order or higher order, you're weak late. And so this shift seems to occur. And so we have a lot more work to do to really study how this shift in strength changes over time. So in the last few moments of the talk, I'm just going to start asking a question which I think immediately comes to mind, having had all this descriptive of it. What would happen if you perturb the afferents, either in terms of their activity or their genetic makeup? Because of course, you can imagine the early sensation is doing some sort of instructive role. So what happens if you take that away? How does it affect the connectivity of these cells? And so we did that in two ways. We did an infraorbital nerve section, which basically cuts all the trigeminal nerves to the barrel field at P0, or we enucleated the animals. We just took away their eyes. And so in either case, that early first order input, which starts, is lost. But of course, later on, you're going to lose higher order input. And we asked, does that loss of sensory activity impact the way interneurons develop? Now, remember, at the beginning of this talk, I told you work from Lopez Bendito that shows that activity has a very strong effect in terms of the way pyramidal cells are innervated. So you might expect that interneurons would follow suit. And in terms of the PV cells, they did. And what's interesting is they don't necessarily lose connectivity, they change what connectivity they get. And so what happens in the interorbital nerve section is now PV cells in S1, when you get rid of the orbital nerve, now get an equal first order and higher order, as compared to control, where it's like a 70-30 split in favor of first order over higher order. However, the SST cells seem to get pretty much the same connectivity, even without sensory activity going to them. And so here's what we see in terms of the connectivity and controls. PV gets a strong first order compared to higher order. But in the infraorbital nerve section, the PVs now get equal first order and higher order. And what's interesting is that is exactly the pattern we see up in M2. And so it's interesting to wonder how this change of activity phenocopies the way PV cells, if they're in M2, get equal first order and higher order versus S1. So the last experiment I'm going to show you today before opening up to questions is what if you do a genetic manipulation? So work by my colleagues David Ginty and Lauren Arfice, noting that there are sensory deficits in various models of autism, looked at how sensory information are changed in these. So of course, one of the caveats to this is when you do a knockout of FMR1 or SHANK-B, the phenotype could be originating in the periphery or on the interneurons. So I'll come back to that. But the important point here is, does it affect them at all? And so the results here, as you can see in this picture here, here's the control for somatostatin. They get equal amounts. But now, when we get rid of FMR1 or SHANK3, they start looking more like the PV cells. And the first orders come in, but that late arrival of the higher order doesn't occur. And so Effectively, it looks like the SST cells are stuck in a developmental state where they really only get the first order and not the higher order. And of course, we'd like to know how this is, what occurs in PV cells, and we're currently looking at that. Now, I mentioned at the beginning of this that you could imagine that cause and effect are mixed up here. Is the genetics affecting the SST cell itself, or is it affecting the sensory information coming up? And the only argument I'll put right here that it may be the former is that I showed you already the SST cell is not affected through a very dramatic effect of sensory information, but is affected by the loss of FMR1. I skipped over it, but SHANK3 has no effect. So again, this is the beginning of what's obviously going to be a much deeper dive into questions of nature and nurture and how interneurons are affected by activity versus genetics. And it's a false dichotomy, as I'm sure many of you know. Activity affects gene expression and gene expression affects activity. So they're going to be hard to tease out. But it really does put a pin on the question of how nature and nurture affects these cells. So I'm just going to summarize what I told you here on the second part of this talk. Presynaptically, local and long-range connectivity is areal specific, independent of cell type. The timing of which they get those connections is cell type dependent, and that the rules of that change depending on what area you look at. And the integration occurs in a discrete temporal order. And finally, what I told you is that thalamocortical activity in terms of first order and higher order is different in PV and SST, both in timing and in type. And so I'm sure that as we dig deeper into that, it'll be super interesting to understand exactly what roles they play in PV and SST circuitry, particularly among different cortical areas. So these are the two areas I talked about, and let me just give credit to the many people. I've more or less given credit as I went along. The first part of this talk was really spearheaded by Kate Alloway, who did all the wet work on it and was really helped by Mariano Gabito, who's a wonderful computational person and has been her partner through that. And some of the early work was done by Orly Wapinski. Finally, the gene regulatory network is really the baby of Giuseppe Salde, who's done a wonderful job working with Mariano, getting that work going. In the second half of my talk, you heard work which is really pioneered by Gabrielle Pouchelon. She was helped by a wonderful undergrad, Andrea Miro, and had work from the Cozart lab from Yannick Bollum. And I'm going to pause there and take any questions that you might have. Thank you.
Okay, thank you very much, Gord. I'm going to ask you to toggle your mic on and off whenever you need to speak. So what's going to happen right now is there's a whole bunch of questions. There's, as you can imagine from this beautiful talk, there's a lot of questions. We'll try to get through as many of them as possible. So what's going to happen, Gord, is I'm going to ask you, I'm going to read the question to you, and then I'll turn my mic off and yours on, and you take it from there, and we'll do that question by question. Okay, so the first question is from Yoram Kaiser. It's actually a compound question because it's a question from Yoram and with comments from Liju and Ayano Matsushima. The basic question is, how do you interpret this decorrelation between expression and chromatin accessibility? What do you think is going on to allow genes that are not chromatin accessible to be expressed and so on? So what explains that correlation molecularly, in your opinion? But also, what are the implications of this lag between the attack-seq and the mRNA, and could some of this be related to regulation by small RNAs that are not necessarily visible in classical mRNA-seq, and obviously not in attack-seq? So I'll turn.
Okay, so I assume my mic is on now. The simple answer to all those questions is yes. The more complex answer is, of course, I don't know, but let me drill down a little bit. how do genes that are not obviously accessible have transcription? And I think that really lies into what do we mean by accessible and not accessible. Clearly, they're somewhat accessible because they wouldn't be turned on if some transcription factor wasn't binding to the enhancer and the promoter and turning them on. So it's clear that what we're saying is inaccessible is not an absolute measure. And really, to understand this, this is just the first pass. And really, the frustrational looking chromatin came from people doing IP, which is on millions of cells. So you're averaging, just like we used to do bulk RNA-seq. Now with single cell, we can look at the accessibility, but at this very crude level. So, you know, there is a wonderful world of people who have looked at histone marks, for instance, and marks on the DNA itself in terms of affecting transcription. And these methods are rapidly getting scaled. So it's not at a single cell level, but we're heading there. So I think as we start getting that information at the histone marks like K27, K4, methylation and acetylation, we're going to get a much better idea of how this regulation occurs. And as a matter of fact, Chenyu Wang, who's a postdoc in my lab, is actually using cut and run methods and trying to do cut and tag methods to start getting at this. But we're just at the beginning of a long path. So the second question is, what is the meaning of the disconnect between chromatin and RNA? What's that telling us about development? And I think the lesson is really, really interesting. My guess, and again, I think we need to test this, is that it says something about the plasticity of the cell. They clearly, with the GRN, segregate more and more as they become more mature. And that segregation is shutting off possibilities to those cells. And that segregation, you bake in identity, I think, at the RNA level, at the chromatin level, you specify identity at the transcriptional level. So I think what's happening is, as they mature, basically the chromatin is getting a structure that allows it to go on kind of autopilot. And I think one of the interesting things which we're looking for is whether genes that we think of as housekeeping genes have very specific effects in PV versus SST cells in the adult, whereas they're generic ones early on. So there's a lot to do, but that's my instinct.
Okay, so the second question is from Elizabeth Ferlanis, asking about how do methylation states look like in chromatin of PV and NSST neurons early on, commenting that recent data from the Ridgely lab showed that early in development, several immediate early genes, but not just those, have a sort of dual state where genes are ready to be expressed, so primed, I guess, but elongation is inhibited. Is it possible that PV and SST cells have similar chromatin openings but different methylation states that would then allow or inhibit gene expression?
Yeah, so that's exactly what I was alluding to, Elizabeth. And I think, frankly, that is a fascinating question that if you wanted to work on, is a super rich area. And as I said, Chenyu Wang is starting to look at that. So I'm sure there's a whole lot of detail, both on the histone and perhaps even on the DNA, in terms of marks that allow you to distinguish that. And the simple answer is, until we get that down to the single cell or at least subtype level, it may be good enough to get it to a subtype level, we're really not going to know the answer. But I think methods are coming online. So I think you have the right instinct, and let's just work a little harder and we're going to figure those questions out.
Okay, so next there's a question from Ayano Matsushima. When exactly was the connectivity assessed in development? And then a follow-up is, even during development, like at E15 versus P5, the connectivity, no. So does the connectivity change even during development if you were to, say, compare E15 to P5? I guess the gist of the question is, when exactly did you assess, and how dynamic is the whole thing?
As I kind of pointed out as I kind of pointed out at the beginning of this, you can imagine how complex and multifactorial this gets. We're looking at different areas, we're looking at different cell types, we're looking at different times. So it's impossible to survey everything. As a matter of fact, I think it's pretty Herculean to look at how much Gabrielle has tackled. So first, I just want to tip my hat to her and the amazing work she's done. Even though rabies is great, and we could talk about the limitations of rabies, strength being the most obvious caveat, that you don't really know anything about strength. It's also limited on when we can get the helper cells. So SST, you're driven by when SST turns on. So you probably could start looking at connectivity with rabies embryonically if you were sufficiently brave to do the injections then. P10 is about as early as we've been able to do it because it's the first time we can selectively mark them. So I think the question of what's happening earlier is great. I just don't think we have the methods to do it yet. And I'm actually kind of amazed we've been able to do it this far. If you told me 10 years ago we'd be here, I would have told you you're out of your mind. So again, I think new methods will come. They always do. But for now, I think Gabrielle's taken this about as deep as you can in terms of how early you can do it. But I think your instinct's right. These things happen earlier and perhaps embryonically, and it would be great to look at.
Okay, so we have a question from our host-in-chief, Denis Jabonneau. Hi, Gord. Any speculation about why circuits have to go through this complex cellular choreography during development and how the area-specific differences identified make sense functionally? Denis, he even signed his questions.
I hope you. So Denis, super interesting idea. I'm going to take a page off Marie Tosca's in thinking about this. What she was showing in a beautiful work she did with Gilles Laurent, she's continuing to work on this on her own, is that interneurons are old. Transcriptionally, interneuron basic types seem to be there in turtle. And again, akin to the work from the Allen, what she discovered was pyramidal cells or projection cells, because they weren't all pyramidal, are more variant than interneuron types, certainly across time and within species themselves. So you have this mismatch between pyramidal cells that are region-specific and area-specific, and there's just gorgeous work from John Rubinstein and Dennis O'Leary and yourself showing the tapestry of how pyramidal cells vary across cortical areas. And so you have a situation where the same interneurons are looking at different pyramidal cells at different times. And they wire differently. I mean, if you go look at an interneuron in the hippocampus and look at one in the cortex, you can see that the wiring is basically shaped to that local area. And the only way you can do that, if they're transcriptionally as similar as they seem to be, would be for a crosstalk between when they get information and how they do it. You can change the way something's wired by doing it in a different order. And so I think by having these mature at different times, you can overcome the fact that they're transcriptionally not that different. It's a first pass. I think there's a lot more to learn, but I think the answer, is that the timing's going to be important, and we're going to learn something about when they get wired in terms of how they wire, and that says there's information transfer, and that gives us a clue where to look.
Okay, we've got a question here from S. Miero, or Miro. Thank you for a fascinating talk. When you referred to PV cells coming from local connectivity early in M2, how early is early? I think you addressed that a little bit earlier. And is this age for forming local connectivity in PV cells and the temporal order relative to SST cells similar in S1 and V1 as in S1 and V1 to M2? So is it in S1 and V1 similar to M2? Is the temporal order similar? Do you want me to repeat that?
Maybe go through an executive summary of it, because there was a lot of, I think I got it, but why don't you reword it and then maybe I'll understand it.
Okay, so, oh, where did it go? It disappeared. Wait.
Yeah.
Got it. I'll read it again. When you refer to PV cells forming local connectivity early in M2, How early is early? That's part one. Part two is this age for forming local connectivity in PV cells and the temporal order relative to SST cells similar in different areas? That is to say, S1, V1 to M2.
As I alluded to earlier, we're limited, particularly in PV cells, how early we can target them. And so that is an analysis done at P10. It's done, obviously as soon as the genetics is there to do the starter cells, and the rabies and the starter cell starts around P5. So you're getting some summation between P5 and P10. And since it's cumulative, all we're seeing is what's at P10. If we want to do an earlier window, we're going to have to shift everything earlier and the analysis, and we can't do that with PV. So in terms of the PV cell, the answer is that's as early as we can look at it. In terms of the timing in different areas, I don't think we quite have the resolution to be super specific about that. We have a limited number of time points. It's basically P10 with the PV and then adults. So that's all we know. And yes, they're at slightly different stages in P10 in M2 versus V1 and S1. Those areas develop at different rates. So that's not terribly surprising. So yes, the timing is going to be a bit different. Is it significant? Is it the same program running, but they're in, we know S1 develops before V1, for instance. Is that all we're looking at? Maybe. Or is there something in the timing that's giving you different information? I think that's the crux of it. And I don't think we know the answer now. And I don't think we quite have the tools. We probably could look later in V1 would be a good thing to do, because since it matures later, the question is that equivalent?
Okay, we've got 4 more questions to go. How do you feel about that? It's okay? So keep going? All right. So the next question is from Amrita Singh, asking, how does timing of interneuron cell type differentiation correlate with the timing of their migration into different regions of the cortex, and does differentiation affect different subtype, the migratory properties of different subtypes? And Shuba Tole wants us to know, and this is pretty impressive actually, that this is a question from her first-year PhD student. It's an excellent question.
So I think anyone who's studied interneurons for a while, and I certainly will excuse a first-year student for not realizing this, knows that they're each born with different temporal windows. So on average, the SST cells are older. They're born, their birth date occurs a little bit earlier than the average PV cell. Now, that's hard to say, right? There's a whole nother level of different types of SST and different types of PV that we're blind to. We're trying to restrict it to deep layers. We're trying to match apples to apples, but there are earlier SST and later born SST cells, and there are slightly different flavors. So in aggregate, SST are born a little earlier than PV. Now, the secondary question is not just when you're born, but when do you get to the cortex and when do you integrate? And that was really nice that you brought that into it. Yes, on average, there's a pretty good correlation to when you're born and when you integrate. So I know some work, I'm not sure it's published or not, by Simon Butt, had shown very nicely that SST signaling matures in deep layers before SST in more superficial layers. And we know that, for instance, layer 1 interneurons are CG derived and born again later. So the question of birth dating is a great one, and it really does get at this temporal element. Now, exactly how it all lines up, not sure, and we need to do more work, but a really, really great question.
Okay, so another question from Ayano asking, subplate cells and their projections or inputs onto them decrease in different speed, I guess decrease at different speeds across S1, V1, and M2. The remaining number of subplates in the remaining number of subplates in the adulthood different, I guess, in these different areas?
So we're getting at what I think is a super interesting question again. You said different speed. Maybe that data is out there. I don't actually have that data. We only looked at a few time points. I think you're probably right. They do regress at different speeds. And in terms of the work here of basically the inputs to PV and SST. I don't know exactly when they disappear. I actually have a graduate student, Shuhan Huang, who is really interested in these subplate cells, mainly in terms of their connections to layer 1, which they also have. And they definitely shift over time. So at the heart of the question is, when do they connect? When is the connection lost? And are there subplate cells in the adult? And so I'll answer those individually. Yes, they have connections. Yes, by and large, connections from subplate to interneurons are lost, as irrespective of what the kind is. The timing, I have no idea. Yes, subplate cells are there in the adult, and some work from Matt Larkham is actually looking at that. So I think one of the things which is kind of in the lore of subplates is that subplate disappears in adults. That is clearly not true. It turns into layer 6B. I'm not even sure how much of the subplate disappears, but they clearly make different connections developmentally in the adult. So I don't think we really understand what these transient changes mean, but it's something I'm really interested in. I think again, you put your finger on something super interesting to look at.
A question from Chris Dalla asking, how do you think about integrating rabies-based connectivity mapping versus synaptic recordings? I assume you can show a connection via rabies tracing with relatively few, maybe weak synapses. I ask because we know that PV cells get more synaptic excitation as they mature, while the rabies tracing suggests maybe the total input doesn't change that much. Local connectivity decreases while contralateral increases. Thanks, great talk.
Okay, but that wasn't Attila Lassange's, was it? Or you don't know?
No, that was Chris Dalla.
I caught that at the beginning and somehow I flipped it. So Chris, this gets into the original rabies was a strain that Ed Callaway used called B19. It was an anatomical tool. It was really not appropriate for using in terms of physiology. It traced okay, but it was super toxic to cells. And actually, I think made interpretation a little difficult because you trace for a while and then you'd start killing both the starter cell and the cells traced to it. Attila Lassange's, that's why I had it in my head, came up with Reardon, came up and Tom Jessel came up with the use of a N2C strain that works perhaps more efficiently and is way less toxic. So Lena Ibrahim in my lab had done the rabies and waited a month, and they're still pretty healthy. So I think actually the kind of question you want to ask about combining rabies with optogenetics is doable. We've started trying that. Gabrielle actually made a flip version of rabies and then used an FDIO channel rhodopsin to target the cells that project into that cell as a way of directly looking at it. The answer is didn't work great. I don't know why, whether that was a technical problem or whether that was something about the rabies, or I don't know. I think the best we can really do is essentially do the optogenetics once we know the connectivity and then assess it. That's of course not a direct apples to apples because you may be hitting a different population than you traced. But some combination of that is needed to be done. As I said, rabies doesn't predict strength, it predicts connectivity. And I know that some people have some doubts about how well rabies works. And what I've heard Ed say, and I totally agree on, is when you see a connection with rabies, it almost uniformly predicts real connections. What it doesn't predict is strength. And the caveat is that certain connectivities are not picked up by rabies. For instance, if you go 2-3 pyramidal cells and look in layer 1, both Ed and I observed that you don't get layer 1 interneurons to 2-3 pyramidal cells, and we know they're there. So I think it's a good technique. I think it has its limitations. I think to answer the question you want is probably best directed by doing a rabies experiment, figure out connectivity, and then do a separate optogenetics experiment to get strength. And they're the kinds of thing we're doing. And certainly I showed you a tidbit of that with the SST. We're hoping to do it with the PV, and that was a lot of your question.
Okay, so we have three more questions because questions were added on, and that's all we're going to do. So please don't add any more questions. You're still okay for three more? Okay. So Francesco Bidoni is asking, "Hi Gord, interesting you focused on Fragile X, where you have hyperconnectivity, hyperexcitability, hyperconnectivity in the cortex. What would happen if you had a hypo situation, such as in MECP2 Rett syndrome models, MECP2 nulls Rett syndrome models?"
Yeah, so the the person who asked the question is clearly more savvy about monogenetic models than I am. I guess my first reaction is, I don't know if I would necessarily say, I mean, you get increased activity in the brain in a Fragile X. The implication often is that you have decreased interneuron function because you're not buffering the excitability as much. Yeah, it doesn't have to be that way. It can be you can imagine it otherwise. But so you asked MECP2. I don't know if I'd classify it necessarily as first and foremost a hypoactivity one. It certainly has lots of effects. I think we're still trying to work it out. And I think the effects are going to be pretty cell type specific. So to me, the question is this, neuropsychiatric disease is a problem of fairly selective brain function. I mean, people who are schizophrenic or bipolar still can get up in the morning and put their clothes on and walk and do stuff. And autistic kids are relatively normal in the first few years of life. Certainly schizophrenic kids are completely normal, and it comes on later. The changes that occur are subtle. And, again, I think what's exciting here is we're tracing developmental events. And I think that as we understand those developmental events and adult functionality better, we can start querying about whether the hypothesis that connectivity is changed and is causal in those is real or not. And I think we're going to have to do it on a case-by-case basis. I think that combinations of global knockouts, which are really the disease, and conditional knockouts, where you can ask about the cell autonomous function of these genes, will start getting at that. I don't think there's a one-stop shopping for this. I think you're going to have to look at a whole bunch of different connectivities and events in those things to really understand the impact of those genes.
Okay, then we have a question from Veronica Moreno Juan. I'll just summarize it in one sentence. When you cut off input, do you see cross-modal changes, or do you affect only the modalities whose input you remove?
I'm sorry, could I missed the end of that.
The question is, when you do IONs in different sensory modalities, are there cross-modal changes in the SST-PV cells, or are they restricted to the parts of the cortex that process that modality?
So I would have to ask Gabrielle. I don't think she particularly looked at the SST or PV cells in V1 and M2 on the infraorbital nerve section to see if there's some knock-on effects. If there is, my guess is it's subtle enough if you're just looking at those cells that it might be hard to pick up. I think there are probably knock-on effects in other cell types. Okay, so she wrote no cross-modality effects. So she actually looked and she did not see it. It was only within the one she looked at. So the ION only affected S1 and the enucleation only affected V1. That's the advantage of Slack. I'm getting real-time information.
Fantastic. Okay, and so the last question is again from Yoram Kaiser. Do you expect interneuron inputs in M1 to be similar to those in M2 or similar to those in S1/V1?
Again, I mean, Gabrielle has done some preliminary look at that, and I can't remember the answer. We certainly haven't done it thoroughly. I think at a certain logic, I'm not surprised that sensory areas in general resemble one another and associational areas are different. I can't remember what the data was for the M1, and I don't think we've done it thoroughly enough that I could make a statement. as a first pass, I bet there's a third variation on how these things work, because primary motor is mainly about output, and associational motor is actually a lot more like prefrontal. So it really is a third category. I think I know I pushed them to look at that, but I don't think we have an answer at this point. And I seem to have lost Bassem, so maybe I've lost everyone. Okay, are you there?
I think I'm back, yes.
I'm back.
I'm back too. Can you see me?
I can't hear you, unfortunately, but I am back.
Yeah, me too.
Okay, let me try rebooting one more time here. Okay.
Hi.
Can you hear me?
Yeah,
Yeah, but I still can't hear you, unfortunately.
that's weird.
Okay, last time this worked, when I quit right out, I'm just going to quit my server and come back in.
Can you hear?
Are you there? I still can't
Yeah,
hear you.
I'm here.
I'm going to leave and come back.
Okay.
I can hear you now.
Okay, so this is just, I just wrote it in the chat box. Thank you everyone. Thank you Gord for a fantastic talk. Tour de force, not surprisingly, wonderful work. You, yeah, once again, I think you're opening a whole area of new exploration. It's going to be, you know, that's going to keep on giving for another 10 years. So we're very excited about that. And thank you everyone for staying late. I'm sorry about the technical problems. We have no idea where they're coming from, but I think we managed just fine. We got through all the questions. Gord, you've been very patient and generous with your time. Thanks very much. And if you have any last thing you want to add,
Yeah,
please go ahead.
Yeah, sure. I would just say I miss the community tremendously. You know, it's nice to get some questions at the end, but I miss the 3 days of wandering around the meeting afterwards to talk to all of you. Stay safe. Science is real. And yeah, hope for good things on November 3rd in terms of the US re-entering the world in a sane way. So peace, safe travels, guys.